decided to sing happy birthday to Luna, and
I
she said she liked it.
I loved it. It was so sweet. I like that birthday song too. I've never heard that birthday song, but I like it. Jenna, I am looking into it. I don't know why yet. I'm still trying to figure out why. So Jenna was asking about so in your getting started stuff, it had you opt in for texting if you're comfortable with that. I love to text via communication because I feel like you all communicate well by text. Like we don't have to text back and forth, but like when I send it through Zoom, it's just like a quick reminder like this is due in the grade book today. If you don't have it done, this is just a reminder to get it done. Like I don't blow up your phone. Who gets texts in here? Does anybody get get text that's on in the class right now in our CC? Anybody get my texts? Well, you should. I have a handful that get texts from me. And I don't blow up your phone. I just wanted somebody to like be a testament of it. That says Mrs. Rittenhouse will not blow up your phone. Okay. It's just a really easy way for me to communicate with you versus so like when we call you, a lot of times it'll come up like OHVA or it'll come up like a corporate number and just quite frankly we get ignored. No one picks up the phone, so I either talk to voicemails, which sometimes works because you all get them and then you're like, oh, it's Mrs. Rittenhouse, I don't have to run, or your voicemails are full so you never know who gets them. Yes, some of you do check emails, but I want to say like maybe 1/6 of you check emails and respond. So it's just an easy way for me to check in with you. You do, Jenna, 100%. A lot of you in the CC do. Um, it's just another way. You should not have it on there twice unless you are in our new cohort. We have a new cohort of about 10 students joining us. So we will do a little orientation at 10:15 for my newbies. So I can get to know you and catch up. Catch up to speed. Yep. Yep, everyone welcome Isaac. Isaac's here. He's part of our new cohort. So he's joining us. We had a bunch of new students joining us in January. But I'm going to be honest with you, this is one of my favorite classes. Like ever. You're going to love this class, Isaac. So I promise you that. I think I'm pretty cool, but the students in this class can also tell you that it is pretty fun. I think they'll enjoy it. You like it here, right, guys? Luna's new this semester, and you would never know it. I feel like I've known Luna forever. It's awesome. Like we've known each other more than a few weeks, right? It feels like we've known each other more than a few weeks. Yes, welcome. Yes, 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 yes. So welcome to class if you're here and it's your first time and you see 2 on your schedule. You won't usually have 2 of me on your schedule, but it's orientation day. So, but before we start into class, Jonathan has a quick question and then we're going to do a quick review from yesterday's content. And then you're going to go take these quiz questions. I'm going to give you about 10 minutes. Yes,
yeah,
Jonathan.
Yes, I do have a question. Like, 2 questions actually. So would it be all right later if I could study this again, just deep inside the lessons?
Absolutely.
Inside.
Yep. You can always study after class. 100%. What else do you got for me, Jonathan?
I, for instance, I had to talk to my teacher because I did her readiness test, but she's still saying I didn't do it or something, or may have an error message like that. I'll have to straighten out with her.
For me?
No, for me, because my math teacher.
Oh, okay. Yep, no big deal. She'll work through that with you.
All right.
So thank you, Jonathan. So what we're going to do right now is we are going to go over, this is the 1.04 quiz questions. Yeah, that's what we're doing. We're literally going to review these, and then you're going to pop into your classroom and take these. I know, Jenna. I'll figure it out. I'm so sorry. Just give me a few minutes or a day or so to figure that out, because I don't know why it's showing that overdue notice. But I
And
know
what
that's
quiz
obnoxious.
question are and what lesson they are we going to do today?
We're doing 1.04, and then we're going to move on to 1.05, 6, and 7. Okay. So let's go ahead. Who would like to read question 1 for our quiz? All right, Luna, go for it.
All right, can y'all hear me?
Yes.
Yeah, thank you guys, by the way. What happens to E. coli when lactose is not present? A, the enzymes. Did I say it right?
Yeah, perfect.
All right, enzymes needed to break down lactose are continually produced. B, the genes that produce the enzymes need to break down lactose are not expressed. C, E. coli is not able to survive without lactose. Or D, the genes required to produce the enzymes that break down lactose are destroyed.
Awesome. So here we go. Go ahead and put in chat what you think. Remember, E. coli is the bacteria that's in our intestines. And we talked about that yesterday about being lactose intolerant and how we have bacteria that help break down the food in our intestines. It was perfect reading for tricky words, too. And that what happens when our body struggles in that, all right? And so, what do you think? Why is that not happening? Yes. So, you are right in answering B. B. The genes. So, the genes, the DNA inside those cells that produce the enzymes needed to break down lactose are not expressed, right? They're not turned on because that repressor is there. And there's people that are lactose intolerant don't produce what they need to lift that repressor. So because that repressor is never lifted, or that gate, it's like that gate on that marble run is never lifted, they aren't able to produce lactose. Good. Who wants to read the second? Lily, you want to read the second question?
When is gene expression blocked in the lac-operon system? Thank you.
Oh, and Gracie, I just got your message. I'm sorry that your polling wasn't working. That's weird. You were right though. The gene expression blocked in the lac-operon system. Is it A, the repressor protein blocks the genes from making mRNA? All right. Is it B, lactose binds to the repressor and changes its shape? RNA polymerase begins transcribing, or the repressor gene destroys the genes that are required to produce the enzyme. When I read questions like this, I really try to lean into some of the key words in there. We never talked about anything getting destroyed. All right. We talked about the repressor blocking things, but we never talked about something getting destroyed. So for me, in my head, that kind of X's out the choice D. All right. We're talking about when gene expression is blocked. What blocked it in that example. I try to think of what words associate. Yes, you're right. Alexis, you're 100% right. We talked about that repressor is what blocks it. So that is what makes me lean into the two answer choices that have the word repressor in it. Repressor and repressor. So that would eliminate C for me. All right, from there, I'm going to go and try to figure out which one is right. Lactose binds to the repressor, changing its shape, or the repressor protein blocks. This is where I'm saying, do I remember anything shape being changed? Do we remember the shape being changed, or do we remember the repressor blocking? What did the repressor do? Did it block or did it change its shape? Yes, the repressor protein blocked. All right, so that helps us choose A. It's okay if you didn't get them all right. First of all, reviewing yesterday's content, which was a little bit, I want to say it was a little bit, it's very, very small detail level in a cell. So it was hard. You guys are doing a great job pulling this stuff back out. Yes, you're right. All right, and last but not least, Nick, will you read our last quick check question from yesterday's stuff?
Yeah. What is the lac operon in E. coli a classic example of? A. Antibiotics blocking protein synthesis. B. Gene expression leading to DNA mutations. C. Gene expressions occurring by changing protein shape. And D. Gene expressions occurring in transcription.
Awesome. High fives to everybody that read. Thank you. All right. Good job reading. I love it. Having your guys' voices on this makes this way more fun. So what do you guys think? What is the lac operon in E. coli an example of? What did this give us an example of last week? Was it antibiotics? Was it gene expression? Is gene expression tied to mutation? Is that what we did? Was it gene expression tied to protein shape? Or was it gene expression tied to transcription? We did, I know. It's a lot of words. All right, we didn't really talk about antibiotics. So if I'm you, I would get rid of antibiotics. We've talked about gene mutations, but we didn't talk about gene mutations being a part of gene expression yesterday. We weren't really talking about mutations, right? We talked about how you have to have the operon, which is the little flag waver saying this is where the DNA starts. And then we had the promoter, right? That either allowed us to make a copy of the DNA, or if there was that repressor on the operator, it blocked us from being able to get a copy. All right. We talked about that in like the banana peel, right? The peel has to come off, just like the repressor has to come off to make that copy. And why are we making copies of DNA? Why are we doing that? To go make something, right? Yeah, to heal our wounds, to make things that we need, to make the proteins that we need. So we're going to get rid of B. So now we're down to protein shape. Did we talk about the protein shape, or did we talk about getting a copy of DNA so that we can take it to the ribosome and make proteins that we need in our bodies through transcription and translation, which is a part of protein synthesis? All right, so the answer would be D. Okay, we're not changing the protein shape. This whole thing of finding the DNA and making a copy is so that we can take it to our ribosomes and make all the proteins that we need for our body to function. Okay? So the answer for that would be D. Okay? So what we're going to do right now is I'm going to give you time to go log in to our biology classroom to open the 1.04 quiz. And you guys can take that right now. Okay? Right now. That's what I want all 100 of you to do, or 99 of you to do, is to log into the classroom, take this quiz. We're being super efficient. We're getting things done. Okay? Nope, that's fine if you were taking it as we went. That's fine too. Remember, what we talked about is unwinding the DNA, finding that little operon that says this is the construction zone, right? Just like there's a sign for everything that we drive by on the highway. Like this is a car wash is here, right? And then we pull up to the car wash, and then there's either a green light or a red light. All right, that is where our promoter is. If the red light is on, we can sit at the front of the car wash all day, but we can't go in until that green light comes on. So when that repressor is lifted, the red light represents the repressor. When that red light is lifted, the repressor or that gate is opened. And it can go in and make a copy of that piece of DNA that it was hoping to make a copy of. That's called transcription. Copying our DNA into a single-stranded piece of RNA is called transcription. It's 1.04. So then we have this single-stranded copy of, you know what, Isaac, you might be exempt from it because you're just starting this week. So you may not see it. Once we have this piece of RNA that we copied in the DNA, it leaves the nucleus, goes out into our ribosomes, which are like a little tape dispenser, right?
Mrs. Rivenhouse.
1 second, Jonathan. You feed that DNA through the ribosome, and the ribosome reads it, right? It matches it up by 3 nucleotides at a time. Who remembers what those 3 nucleotides are called? And it codes for an amino acid. 3 nucleotides at a time. 3 letters at a time. What are those called? The DNA sequence or the RNA sequence, 3 at a time, it gets read through the ribosome, like CTA. You're right. So it could read CTA. It starts with a letter C. 3 of those are called a what? C? Nope, we're gonna keep going today. We just are gonna do the quiz first. CO. Yes, Darian, it's a codon. Okay, that is a codon. So our messenger RNA goes through the ribosome and reads them 3 letters at a time. Those are called codons. Yep, no worries. All right, and that is how they make proteins. They string those 3 nucleotide codon. It codes for a amino acid bead that we put on a strand. Strands of amino acids make what? I know it's a lot this morning. Strands of amino acids, yes, Alexis, make proteins. Yep. Good. And good job, Mila. I see you working hard over there. All right. So let's turn into a new chapter. Everybody write those down if you haven't written those down yet. We are going to go into 1.06, 1.07, which is biotechnology. It's super fun. All right. No. Here we go. So look, what color are we looking at today? We're taking what we've learned this week so far, and we're moving up the triangle. All right. We're going down here to some analysis and some understanding concepts. Okay. So we're going to take what we call recombinant DNA. We don't necessarily know what recombinant DNA is, but we know what DNA is, right? Oh, Sorry, guys. Can you hear me now? Okay. So recombinant DNA, we don't know what it is, but we all know what DNA is. What would you tell me? Let's just, before we even start, let's go through the word recombinant. What do you have to guess about the word recombinant? We could slice and dice this word. Let's dissect it. Good. Good, James. We could say recombine. All right. What does the word combine mean? What does combine mean? Put together. So we're going to re-put together DNA. What does that sound like? Does that sound trippy or what? Yeah. Put back together, join together. Sounds funky. Yeah. Sounds like we're about to mess up some DNA. And you're right in saying that. All right. So we're going to look right now at what is recombinant DNA. Once we figure that out, we're going to really process what that means today. We're going to understand it and we're going to analyze, like, what does that mean? Like, do we like this? Do we not like this? All right. Biotechnology is a science that we use that involves living organisms to produce needed materials. Using living organisms to produce needed materials. All right. Yep. That is the DNA. And what they're doing is, like, with machines is kind of what we're going to be doing, but on a molecular level. So genetic engineering is a process that involves identifying one organism and transferring it to another. All right. Usually this is to get, like, pass on a characteristic that is new, that the original organism doesn't have. Okay. So let's look at this chart. Genetic engineering is a part of biotechnology. All right. It benefits who? Humanity. It does benefit us. But it also creates somewhat. What is controversy? What is controversy? It says that is yes. Drama. Disagreement, right? Sides. I like all of those. Yes. So there are different sides that we can. So this be kind of I like to say this becomes like an ethical issue, right? Yes, it's controversial to eat. I love that, Gracie. Pineapple on pizza, right? Like, some people would say that's messed up. Other people would say it's delicious. I think it's delicious. Okay. Yes. Dun dun dun. Genetic engineering also creates recombinant DNA and new genes, new proteins, and new traits. So let's just dive into this head first. All right. Recombinant DNA is usually found in the role of a plasmid. Everybody say plasmid. I mean, it just sounds like a biotechnology genetic engineering word. Plasmids. All right. I want you to know this about plasmids. I want you to put this in chat. Plasmids, type out plasmids, equals circular DNA. Okay. I want you to know this. This is stuff that could be on your test in the spring for the state. Plasmids are important for genetic engineering because they are round, circular pieces of DNA. All right. They are found in bacteria. I mean, because would we want to use necessarily a human to produce all the things that we need in a lab? I mean, that might be kind of weird. We could. But we can grow bacteria in a lab and they will reproduce on their own. And so we use these circular pieces of DNA called plasmids as a vector. All right, that means they are, a vector is down here. I'm going to highlight the word for you. All right, as vectors. All right, vectors are where we take DNA and integrate it into other DNA. And the way or the vehicle we use for that is called a plasmid. All right. A vehicle, I want you to think about it, is basically like if I ordered yesterday or a couple days ago, I ordered a charging dock station for our electrical devices, right? One of those things that has like multiple plugs in it for all of our tablets and phones and stuff. I wanted to get that to my house. So the vehicle to get that package to my house was an Amazon van. All right, a vector is a way that we transfer DNA, right? So a vector is the Amazon Prime van, and the package that it gets here in is the plasmid. Does that make sense? The plasmid is the DNA that goes from one place to another. And how it does that is in a vector, like the Amazon Prime van. All right, so this is what happens is we use DNA pieces that we like. I love this illustration because it literally shows you that segment of DNA. All right. So here's the plasmid. All right. We digest it with an enzyme, and an enzyme, what that enzyme does is it acts as the scissors. All right. And it cuts out the piece of DNA that we were wanting to use. In this place, we're looking at like insulin. It's saying, I want this piece of insulin. So we put an enzyme in that cell, into that DNA that will cut out that piece. We then take that piece and put it into another plasmid cell and join it. All right, so there's one enzyme that is the cutter, another enzyme that is like legitimately like a glue stick. Its job is to go in there and stick them together. So we actually have little circular pieces of DNA that we cut and paste, just like paper. Okay? And that is what we call recombinant DNA. Does that kind of make sense? Does that kind of make sense? There are circular pieces of DNA. And we put in enzymes that we know that will go in there and cut a segment out that we want. Then we transfer that piece to another dish with a plasmid piece of DNA and an inject an enzyme that will glue it together into that piece. Yep, I know, we're on a schedule, I promise. Okay, good. You're following me with this. We've talked about this before, but this is what we call an electrophoresis tank. So we can take a piece of DNA and put it in this electrophoresis tank. And what it does is they all start down here. All the DNA starts down here in the wells. And then as we run the electric current through this, what it does is it separates the DNA into pieces. It's really super cool. It breaks up that DNA into pieces and it ends up looking like this. So these are those wells. And when you run the current through the water, it breaks up the DNA into all of these pieces. into these fragments. All right. And so that's kind of how we get these pieces of DNA that we can experiment with or cut and paste from. Right. So the heaviest moving fragments get stuck behind here, where the lighter fragments get separated down here. Does that make sense? So the lighter fragments move faster, and the slower ones get left behind further. And what that does is it just separates out our DNA so that we can look at it in more closely in pieces. All right. And everyone's DNA would look different. So if we did this with yours and mine, they would fragment out differently. Now, if I took two cells of mine and put them in different wells, then they would fragment out, they would electrophoresis out, and they would look the same. So if you're like looking for something the same on your state test, look at like well number 1 and well number 4. See how their fragmentation is the same? So you would, if this was a test question for the end of the year, and this is not in a living person, we'd take cells off of a person. All right, we digest the cell membrane and pull out the nucleus, and then it would fragment these DNA pieces out. So if it's asking you in this, which two are the same organism or came from the same organism, you would look for 2 lanes in the gel electrophoresis that look the same. Does that make sense? If you're looking at a gel electrophoresis, you're going to look for 2 lanes that have the DNA that sorted out the same. And that would mean they came from the same organism. They have the same DNA. Okay. Good. You're following me here. So let's do this. Let's quick check and end here. Which phrase best defines biotechnology? I'm going to just do these today because we're going to end here quickly. Is it a science involving living organisms needed to produce materials? The practice of using technology in science experiments, or the use of technology in solving medical problems? It's tricky. This is a lot. It's actually going to be, I actually don't love this question. And neither do you guys. We have a tie between A and C. Why do you think there's a tie between A and C? And which do you think personally is the better answer? What do you think? If I were taking that, that would be what trips me up too, because I think those are both the same things. Let's think about why we use biotechnology. Why would we do this if it's controversial? Why would we do this if we think there's ethical concerns? Would we just do it for fun? Would we be like, oh, this just looks like a fun, weird thing to do. Let's just stir a bunch of people up, screw with our heads, really just try to see if we can get ourselves into a mess? Is that what we would do? I don't think so. I don't think so. So we do biotechnology, like for that example with the plasmids, we do it because we want to make medical advancements to help life. Okay? We want to do medical advancements that help us in general in life. So the answer to that would be what? I'm going to re-poll you. So I really do want you to think about these. These are questions you can ask yourself. This is a test prep strategy. All right. What do you think? Sorry, I'm making sure this is open for today. What do you guys think? It's going to be the science that involves using living organisms to produce needed materials. All right, because it is needed and because of living organisms, that's why this would be the best answer. Okay. Which tool in biotechnologies do investigators use when trying to connect a suspect with a crime? Which one? Sending a poll. What do you think? It is DNA fingerprinting. Yep. That was a quick one. You guys responded quickly to that. What about the substance? Which substance will a bacterium produce when the human gene is added to its genome? Carbohydrates, a protein made of human and bacterial properties, human plasmids that can be isolated from the bacterium, or the human code or the human protein coded for the human gene. What do you think? This is going to be the human protein. This sounds weird, but that's what it's going to be. This one right here. So basically, there's an indicator of humanity, right? And when that is in any kind of DNA, that proteins for human life will be produced, and that's going to be an indicator. All right, that's a tricky, weird question. We didn't get super into that today. So what I want you to do is make note of these answers before you leave. We're at 9:09. You have your next class at 9:15, I believe. Okay, so we're doing good on time. Note these. Guys, we've covered some thick stuff today in the last 2 days. So what we're going to do tomorrow is make sure we feel good with this content. I want you to level up today. There is a biotechnology quiz. All right, so I want you to take that today. Take that now. If you have time between class, you still should have your class pulled up. So what I want you to do is go in. Go take that quiz today. Take it right now. It's going to be due by lunchtime today, right? I'm putting the due date in. It'll be due by lunch. I want you to go and take this before it escapes your mind, okay? I don't want it to escape your brain before you come back and take this. Write down what you need to take and level up today. All right, so we should have at this point 3 quizzes taken: 102, 104, 106. 102, 104, 106. You got this, guys. Sorry, I had to power through some of that today. If you have questions, stick around.